Hello and welcome to this quick review looking at amino acids, amides and chirality. It's designed to be used with your independent study materials and notes that you've already done, so you need to have them with you while you're watching it. And we'll also focus on amino acid structure reactions and stereoisomerism. And it should follow some work or revision that you've already done on amines, because during the course of the clip we'll assume that some amine chemistry is sort of sufficient and comfortable in your mind, first of all. So if you're not sure about amine chemistry, it might be worth going back and having a look at that first. So just to give you a little bit of background as to what an amino acid is, from a biologist's point of view, they're seen as the building blocks of life because they make up proteins that in turn make up many of the soft tissues in our bodies, such as muscle. It's thought that they first formed on Earth way back in the era of the primordial soup, which is a term used to describe the sort of mixture of different gases that existed in our atmosphere at the time, in particular methane as a source of carbon and hydrogen, and ammonia as a source of nitrogen, as well as water vapour. It's thought to be 20 naturally known amino acids, which make up the proteins that compose our bodies, including things like DNA, and then other things like actin and myosin found in muscle fibres. However, from a chemistry point of view, an amino acid has two interesting functional groups. It's got carboxylic acid at one end, and it's got an amine group at the other. And each of those can exhibit all of the chemistry and chemical reactions of the carboxylic acids, in the case of the carboxyl group, and of the amines, in the case of the amine group. So the R group can be any aliphatic or aromatic structure. The carbon atom adjacent to the carbon on the COOH group is called the alpha carbon. So this structure represents the template for all alpha amino acids. So we've got some examples here of alpha amino acids. You can see that they have the systematic name and also a slightly shortened name that biologists may be more familiar with, such as alanine or glutamic acid or serine, for example. So here's an exercise for you to try. In the box I've given you 3 tasks to complete. So pause the clip and try them, thinking very carefully about mole ratios as well as the products themselves. You may need to go back and have a look at some of your notes on carboxylic acids or some of your notes on amines to familiarise yourself again. So hopefully you got some answers like that. If you got them exactly as I've written them down, then you're doing really well, because I made these a little bit tricky on purpose. So staying with the questions and tasks to do, pause the clip again and see if you can predict the bond angles at these locations in an alpha amino acid. So pause the clip again and use the diagram on the left to help you visualise and recall some of your work on shapes from your first year. You may need to dig out your flashcards with the different bond angles, but hopefully you might be able to predict them from memory. So hopefully you got these ones. It's pretty much all the different bond angles you come across apart from 90 degrees. So this would be a typical example of a synoptic question connecting different topics together that may seem unrelated in your mind, but in fact they're all part of the same thing really, chemistry, if you think about it. And across all your topics it's a good idea to expect this type of challenge in your final A-level papers, particularly the new specification papers. So to summarise the chemical reactions that we've looked at so far, in terms of the acid-base reactions of amino acids, the carboxyl group behaves as a carboxylic acid independently of the rest of the molecule, and then the NH2 group behaves as a base and an amine independently of the rest of the molecule. So it shouldn't be surprising to find that you can esterify amino acids by getting the carboxylic acid group independently to react with alcohols as if it was just a normal carboxylic acid, which in this reaction it essentially is. So looking at the equation, you can see that the amine group is left alone, and the R group is left alone, and the alpha carbon is left alone. All that happens is the acidic carboxylic acid group is esterified, but any other functional groups remain unchanged. So looking at the reactions of the amine group with acids, the amine group will react with them in exactly the same way as normal amines would to make salts, like you can see from the equation. But this time the carboxylic acid group remains unchanged, it doesn't react, because you're reacting the molecule with an acid, and a carboxylic acid wouldn't react with another acid. So, a new functional group, amides, are formed when amines and acyl chlorides react with each other via a nucleophilic addition-elimination mechanism. You may have come across this reaction before. I've stuck it down in red just to remind you. And all an amide is a functional group that contains a carbon double bond oxygen with an amine group of some kind directly bonded to the carbon on C double bond O. So it can be primary, secondary, tertiary, or N substituted. Let me give you an example of each. So using the guidelines at the bottom right-hand corner of the screen, see if you can predict what the names of the amides A, B, and C might be. Pause the clip and see what you can come up with. So the first one should have been pretty easy, because you can just base it on what I've put for step 1 for naming an amide. So the second one, again, not too difficult, N-methyl propanamide. But the third one I've made a bit harder because I've double substituted onto the nitrogen. So this time, remembering alphabetical order for the groups, N-ethyl, N-methyl, phenylamide. Or you could say N-ethyl, N-methyl, phenylamide. Either one of the two would be fine in terms of pronunciation. But obviously your exam is not a pronunciation and spoken paper. It's a written one, so it doesn't really matter. And in terms of classification, ethanamide is primary, N-methyl propanamide is secondary, and N-ethyl, N-methyl phenylamide is tertiary. So again, it's based on the number of R groups coming off the nitrogen, just like it was with amines. So in the final part of this clip, we'll have a quick look at chirality. So if you draw out molecules in 3 dimensions, you can often see that there are 4 different groups around a central carbon atom. So this central carbon is called the chiral carbon, and there may be more than one in a molecule. The 2 optical isomers rotate plane polarized light in different directions. And the structures are non-superimposable mirror images of each other. This idea plays an important role in biologically active molecules such as enzymes and pharmaceutical drugs. One isomer may be ineffective or have toxic effects, whilst the other has the desired biological function in the body. Now if you have a look at the structure of an amino acid I've left there, an alpha amino acid in particular, so looking at the chiral carbon within alpha amino acids, there's no coincidence in this fact, because these amino acids have essential importance in biochemistry and the pathways used by the body in many, many species to produce biologically important molecules. And just to illustrate the importance of chirality in pharmaceuticals, you may have heard of a drug called thalidomide. There was a major scandal in the 1950s and 1960s when this drug was widely prescribed for morning sickness in pregnant women. One isomer cured the morning sickness, but the other caused birth defects, in particular shortened arms. It took a very long time for the drug company that developed this drug to admit culpability. So nowadays, drug companies spend a lot of time ensuring that the correct chiral isomer, known as an enantiomer, ends up in their final product. So the way they do this is by isolating the starting molecule that they want from the plant or microorganism that secretes it or produces it. And because it's evolved over millions of years, it'll be the correct enantiomer to have the appropriate biological effect. What this allows them to do is then use this to make the precursor molecule, or through a number of stages, end up with a precursor molecule which is more quickly made, so it costs less from the drug company's point of view. And then finally, when they get their target molecule, or the final drug, it's safe and effective, meaning it's a more effective dosage, so the same amount has a greater beneficial pharmaceutical effect, and it's also less toxic side effects for the patient. So what this does is it removes the need for the drug company to go through the expensive process of separating out the wrong enantiomer, the one that might be dangerous or the one that might be ineffective. So it's a win-win situation. It's more effective and more cost-effective, rather, for the drug company, and it's also safer for the end user, the patient that has to take the drug. So this is how they do it these days to try and avoid the problems that were brought to light by the thalidomide scandal. Okay, so hopefully this has been a fairly useful review of amino acids connecting to amines, amides, and a little bit of a look at chirality and the implications for pharmacology, pharmacy and medicine. So until next time, make sure you've done a bit of practice on some of the questions on this topic. But thanks for your time, and I'll see you soon.